second video I want to do for you guys today has to do with the basics of the viral life cycle. So again, if we were talking about specific viruses like HIV versus the influenza virus versus varicella zoster, there would be some very important distinct details in each of those life cycles, but this is the general steps that every virus has to go through. So the first one, so here I have my host cell, and then for part of this, I've drawn a naked virus, a nucleocapsid portion, and then I've also drawn an enveloped virus because there's some differences in terms of exactly what happens with those. The very first step is attachment. A big part of attachment is recognition of the host cell. Many viruses have only very specific types of host cells that they can actually invade and replicate inside of. And so attachment is often a very specific process. It often involves interactions between proteins on the viral surface and then receptors or proteins on the host cell surface. And so for either of these viruses, we can have an interaction here where the virus has now successfully recognized our host cell and is attached. Okay? Now after attachment, what we have to have is we have to have entry of a portion of the virus. Now, in terms of entry, different viruses do different things. So for some viruses, the entire virus will actually enter, and then for others, only the nucleocapsid portion will enter, and for others, only the genetic information will enter. So we can have some variation there, okay? The way that the virus enters determines whether the next step actually has to happen or not. Because the next step is uncoding of the genetic information. And so if either the entire virus or the nucleocapsid is what enters into the host cell, then that genetic information has to be uncovered so that the host cell machinery can work with it. So, our three possibilities for these two steps, one is that just the genetic information from the virus would enter. This happens very frequently with bacteriophages. They tend to just inject their DNA into the host cell. In that case, this genetic information can be acted upon. There's no uncoding step that's necessary. But another possibility would be that the entire nucleocapsid would enter, and in that case, the capsid part would have to fall apart for the genetic information to be uncoded. You also, in a case with an enveloped virus, one thing that can commonly happen is that the viral envelope will actually fuse with the host cell membrane. And so then the nucleocapsid would enter, and then it would also have to be uncoded in order for the host cell machinery to work on it. One thing to note here that's important in other classes is that in this case, a lot of times a portion of the viral envelope will actually stay on the host cell surface. And this means that there are some viral proteins that are on the outside of the host cell for a period of time. This can make this host cell targeted by the immune response. Okay? But regardless of how entry happened and whether or not uncoding was necessary, at this point in time, we have the viral genetic information that the host cell machinery can work on. And so step 4 in this is synthesis. And so now what's going to happen is we're going to get gene expression of the viral genome. So it's going to be transcribed and translated. We're going to get viral proteins. We're going to get new viral genome. And basically we're going to have a whole bunch of pieces of virus during the synthesis portion. And so the 5th step is assembly. We're going to take all those pieces and we're going to reassemble the virus. So we might have our genetic information. We're going to have that inside of a capsid now. And we're going to have the virus assembled. And then the last step is release. This is where the new virus gets released from the host cell. This can either happen through a process called budding, where the virus blebs off from the host cell, or it can happen due to lysis of the host cell, where the entire host cell is destroyed. In the event that we're talking about an enveloped virus, one thing that will often happen with these is they will sort of steal a portion of the host cell membrane as they leave, And that can be a big part of how they end up with their envelope. And so again, the details here, where portions of synthesis and assembly happen, how viruses enter, what they recognize for attachment, whether they have encoding or not, all of that can differ with different viruses, but the 6 basic steps are the same.